Okay, so hi, my name is Daniel Wood from the University of Sheffield, and I'm going to be telling you about the convergent gene recruitment and the evolution of aquatic cam in eudicots and lycopods. So living things have got all sorts of complex traits in them. So things like this bacterial flagellum have lots of different components that all come together to produce a specific function, so motility, for example. And how do these things come about through evolution? Well, in many cases, it's that the components are actually just recruited from something that was already there. So in the case of the bacterial flagellum, people think that this type 3 secretion system that was already there, a lot of those components were recruited into the bacterial flagellum. So how restricted is the pool of components available for recruitment to a given new function? So clearly some components are going to be suitable to be recruited into a new function, and others may be less so. So I want to know though, does this extend to genes within the same gene lineage? So there are also genes, so for example, malate dehydrogenase, that are present in about, so say in plants, 4 copies in the genomes. They've been duplicated over time. They're all pretty similar in sequence, but there are differences in expression and slight differences in their sequences. And I want to know whether they're all equally suitable for being recruited to new traits, or whether some of them are really suitable and others recruitment would be a bad idea. So the important thing, I think, is to look at this in different genomic backgrounds, because it's very well to say that in genome A, one particular gene is really suitable for being recruited into a new function, but in genome B, it might not be the case. It might be that genome B doesn't have that particular member of the gene family. It might be that it does have it, but it's being used for a different function which is incompatible with that new function. It might be that there's an epistatic interaction, so something else in the genome is telling you don't use that particular allele. So it might be that that allele, or not that allele, sorry, that member of the gene lineage isn't being used for that function. So maybe a different one is being used instead. Or maybe you just can't evolve that trait at all. So it kind of plays into how evolvable traits are, and how it depends on maybe slight differences in which gene family members you've got. So I want to look at some very different genomes and ask, do they evolve the same trait using the same members within a gene family with the same specific lineages? So the very different genomes I've been looking at are Isweeties and Litorella. So they look pretty similar. They're both aquatic plants. Isweeties is the slightly bigger one at the back, Litorella's in the front. They both live in the same environment, so in these low nutrient, low carbon lakes, like this one's in Wales, where we've got these plants. They both actually diverged 300 million years ago, so they look really similar, but actually their genomes are very distantly related. They're no more related than this man and this salmon. So, despite those differences, both of them have evolved crassulacean acid metabolism, or CAM, which is a CO2 concentration mechanism which basically works by CO2 being taken up at night, and then it's fixed to a 3-carbon acid to make a 4-carbon acid, that's stored in the vacuole, and then in the day the CO2 is re-released and incorporated into the normal C3 cycle, and that allows them to survive in these low CO2 environments because it allows them to sort of double the amount of CO2 they can get, so they can take it up at night as well as during the day. And the good thing about CAM for these sorts of questions is that it involves a lot of genes, so all the genes involved in the catalytic steps here, but then also transporters and regulatory ones which control the circadian rhythm. And crucially, all of the ones we know about are recruited from genes already present in non-CAM plants. So we've got this really similar adaptation, not just CAM, but CAM specifically optimized for, underwater, low nutrient environments in these 2 species which live in the same environment, even though their genomes are super divergent. So were the same gene family members recruited for CAM in Isweeties and Litorella or not? So we used RNA sequencing to identify the CAM gene families. So we did RNA-seq, we built phylogenies of all the genes and looked at their expression levels. And that's just a diagram of the CAM cycle. I've got the green is Litorella and the blue is Isweeties. We won't ignore that too much. The basic take-home conclusion is that there is broadly similar expression patterns for the core CAM genes. And in particular, for the 5 genes which I've identified so far, where there's more than one lineage detected in my transcriptomes, in 4 out of the 5 cases, Isweeties and Litorella share the most highly expressed gene. So the panel on the left is beta carbonic anhydrase, and you can see that green, the Litorella, is expressing the BCA1 more highly, but the Isweeties in blue is expressing BCA2 more highly. But in those other cases, so with PMDH, you hopefully see the Isweeties and Litorella are both most highly expressing PMDH2 as opposed to PMDH1. there we are. So, despite 300 million years of divergence, they've recruited similar gene family members for CAM.